first slide the title slide I had shared with you, right? Could you just could you just note the title, right? Could you note the title? Hello? I hope.
Yes, ma'am.
Yeah. What was it about?
Unculturable bacteria.
Unculturable bacteria. Have you heard this term before? Okay, not really. Fine, doesn't matter. So the thing is, when we talk about, say, bacterial cultures, the word comes as culture, right? And bacteria is to microbiology when we talk in context with that. It was almost 3 centuries ago when studies in microbiology had begun, 18th century, right? So microbiologists had started obtaining the pure cultures, and what as we do today, it was same like the technique for plating technique, isolation by plating technique that was developed, and traditionally we are using that. Further, culture dependent methods, when we talk about, we need certain information, right, which requires the cultivation of microorganism. For that, uh, from which source we are isolating it and what kind of its requirement would be, will it grow onto a particular media or we need a different media. This kind of information we have today. A lot many different medias have come up, yet we have failed to explore the diversity of microorganisms. Are you aware, though the microbial diversity on earth is so rich, out of which more than 99% of species are undiscovered even today, and very small fraction of this 1% which is discovered is culturable. So you can really grow them in the laboratory by available culture techniques. All right. So microbial availability by cultivation has created an emerging need, you can say, to learn more about the missing species and their functions, which are going to help us not only to know about their microbial processes and functions and the information about the organism, but the role of the organism in human existence, right? So environmental sustainability, when it comes, role of microbes is immense, and they are the ultimate game players, right? We all know. So bioremediation, bio waste industries, all these things are very much in a search of bacterial species or microbial species which can degrade this, bioremediate the polluted sites, and keep the environment clean, right? However, though there are so many organisms, we are not able to explore uh, uh, the diversity available that we all know. So environmental biotechnology suffers because of the limitations of culturing technique, right? So do we have any other option? How can we go about it? And so many things to be talked about when we talk about unculturable bacteria. The word tells you it is unculturable. Are bacteria unculturable really, or we do fail to culture them? What is the truth? What is the real story? So of course, bacteria are cultivated in the laboratory conditions, but there are certain bacteria which are not able to grow in standard laboratory media. Why it is happening? What are the reasons? Can we have a discussion on that? Right? And can we really see to it that these species which are not coming or growing onto laboratory media are made to grow into the laboratory media. All right. So let's talk about these in the today's session. Now, when I say about Unculturable bacteria. Just I'm sharing the slides. Yeah, fine. Now, when I say this, the microorganisms, we know they are available, omnipresent, they are, right? So, different environmental sources, water, soil, air, everywhere, and also animal microbiomes are there. But we need to explore them. So, when we talk about the potentiality of traditional culturing methods, it is too limited. And so many, more than 99%, so about 10 raised to 12 microbial species, about they remain undiscovered. So what can we do? Generally, when we approach, generally when we approach isolation of an organism or attempt to grow it in the laboratory, as it was done by Cox. Cox postulates, you remember, right? What do Cox postulates say? Ki the organism should be grown in the plate and it should be able to um cause the disease again when it is put back into the source organism. With respect to medical microbiology, we talk about Cox postulates, but is it limited to that? Is it not giving us lot much of information about that bacteria, its processes, its interactions, its pathways, its selectivity on for growth in culture medias, and so on and so forth? So all these aspects are to be talked when we say about unculturable bacteria. We are not able to culture them. It doesn't mean that they do not grow, right? So let's see our the way we attempt any isolation process. Generally, we go for solid plate media for cultivation of these organisms, and sometimes we go for selective enrichment in the liquid media. All right. So, for example, we are approaching to isolate bacteria from a soil sample or from a marine water sample, right? How do we go? Let's take the example of soil. We go generally for 10 gram of soil samples which are pulverized with mortar and pestle. Then they are dried at room temperature. Then we generally go for its heating for 15 minutes at 55 degrees Celsius. This is for what? If there are spore formers, they will be uh, enriched, right? Because many of the bacteria they go into spore formation mode if they are facing starvation conditions, if they are challenged by environmental parameters. So if you want to enrich spore forming bacteria, you have to go for 15 minutes treatment at 55 degrees Celsius, right? Now, the suspensions then we are going to prepare, and these cell suspensions, I'm sorry.
बघितलं मी पण
Hello, please
तुम्हीच
keep your
सांगा
voice off. Cross connection लगता है. Right. Thank you. So, cell suspensions you are going to prepare 1 gram of soil. You are going to take in a known volume, say 10 ml of the sterile deionized water, and you are going to keep the suspension ready. Now you are going to prepare the dilutions, and you are going to plate it out. Right. Now, after incubation, generally 24 hours or 48 hours, you are going to go for scoring the growth. Now, how do you go for scoring the growth? You are going to count the bacterial colonies growing in a plate. Imagine in place of plate, if you are going with micro titer plates, right? 96 well micro titer plate, if you are inoculated in that, right? So you need to observe that microtiter plate under microscope, dissecting microscope, which is going to give you about 50 times magnification range, and you are going to note down the number of colonies that are appearing. So microtiter plate, you know the dimensions of the microtiter plate, how small they are, what is their area, and how much media they can hold. You have seen those plates, right? So instead of culturing your bacteria in plate, you are culturing it in a microtiter plate, 96 well plate. And then after microscopy, if you observe that there are some colonies growing, you are going to note down the time and after how many hours they have appeared. Of course, it's not that always you will get the colonies within 24 to 48 hours, but it may take weeks together to see the colonies if the samples are obtained from natural environmental sources, right? Where the number of bacteria present in that particular mm, niche will be immense number, immense diversity. But the volume that you are going to bring to your laboratory, how many cells of that bacteria per unit volume would be there, right? Even though there are lakhs of them, how much will come into the water sample that you are collecting or soil sample that you are collecting, and how many different types would be present in a given sample? These are the questions, right? Now, generally when we go for isolation, another thing that we do these days is DNA isolation. We do of the isolates, and then we go for 16S rRNA sequencing of the chromosomal DNA that is isolated from these bacterial cells, and then for which you of course take help of PCR based amplification of 16S rRNA gene, and then you go for GOTAC, green master mix, and then different primers, uh, forward primer and reverse primer they are used, and then you are going to process your samples for amplification, and the amplified product of PCR thermocycler, that you run it for specified number of cycles, say 30 cycles for example, and then the amplified product of the DNA that you are going to sequence. So sequencers are there, different types of sequencing machines are there, automated sequencer. For example, the name I have highlighted here is Macrogen. Of course, next generation sequencing tools, they are continuously advancing, and you have a n number of different methods, improved ways of sequencing bacterial genomes. So once auto sequencer gives its data, that data is going to be a long chain of sequence, polynucleotide sequence, that you have to put to another software tool, wherein you are going to match it with the existing data. So library is there. In the library, genomic data of n number of bacteria isolated from n number of diverse environmental sources are already deposited. You have to select the source, you have to select the type of organism, you have to select the sequence that you have you have obtained from sequencing, and you have to blast it. That means you have to compare it. For comparison, we are not going to do it. You have to just upload it to the library and then say blast. It will be done by the software tools, and you will get the data that will talk about what is the similarity, percent similarity, or sequence similarity. uh, it will be comparing the data with operational taxonomic units, OTUs, that is species, right? So it will be comparing it with that, and will tell you whether there is 99% similarity, 97% similarity, 95%, 60%, or no similarity at all also happen. So if there is very good similarity like 95 to 99%, then chances are there the organism is already known and belongs to the species whose DNA is matching with it for 99%. Right? So these are the different methods, sorry, these are the different steps in a method when you are going to check for the presence of a particular bacteria in a given sample. This is what a part of it that you routinely do. That is, you take the sample, you process it, you put it in the enrichment media, or you put it in the solid plate media and grow it, and find the colonies and go by biochemical analysis methods, right? But it has got limitations. So advanced step is DNA isolation, is sequencing, and you are going to sequence similarity cutoffs. You are going to find out, and you are going to know the uh, name of the particular bacterial genus, species, whether it is already known or it is a new one. All right. Now, this is one way of doing it. But do you think the number of colonies which are appearing in your plate after what we say isolation and plating and incubation are going to be exactly the number of bacteria present in a given sample? If you try to take the sample, put it under microscope and observe it, you will see various methods as against that of the one which is growing in the agar plate. So in agar plate, you may find one or two colonies as against tens and hundreds found under microscope. So what happened to all other 99% of them? They were seen under microscope. They are bacteria. Why are they not appearing in the agar plate? Right? Even if you do sequencing of isolates, you will get to know their genomes. You will find their sequence similarities. You will try to identify them. But what about all those remaining 99%? How are we going to find them out? Under microscope, they are seen. Why are they not growing onto the plate? So this um, conclusion, as it is shown in this particular slide, that under microscope you are seeing a rich diversity of bacterial forms as against very limited. There is 1/100th of it, maybe, or even less, is seen when you are trying to grow them on the plate. This is termed as the great plate count anomaly. What we say कि these many bacteria are present as so many colonies are produced, and if there are of different types, these different types are there. That is how you talk about and conclude your experiment, right? But is it really true? SPC आप करते हो, right? या TVC करते हो, that gives you the results. But is it really representing the diversity of a given sample? Of course not. Under limited conditions, that is a type of nutrient media you have chosen, incubation time, the pH and other abiotic factors which contribute to selection of certain bacteria which are growing fast and forming the colonies. And you count those and you say this is the TVC. Right? So the concern here is the great uh, uh, plate count anomaly is exact number, and the number that appears in the plate is not same. Right? What you encounter, what you find under microscope and that is found under on the plate are totally different. Right? So this phenomena of plate count anomaly was talked as early as in 1985 by Stanley and Konopaka. So the scientists have coined this. This is a common observation. We also note that, right? So how are we going to proceed with it? How are we going to deal with it? Is it about only the environmental samples? What about condition, like a person is suffering from a particular disease? So if it is caused by a bacteria, lot many copies of that bacteria will be inhabiting the body of that organic animal, may it be human being. And then if you try to attempt to take the sample and put it on the media, will it grow or not? Has to grow, right? That is how you find the bacteria from pus sample, or maybe from stool sample or urine sample, etc., etc. But you will be shocked to know that certain bacteria remained unculturable even in the laboratory. For example, Mycobacterium leprae, you must have heard about it, right? So when we talk about Mycobacterium leprae, it was uh, it remained unculturable for a very long time. So why it would happen? Uh, is it not there present in the host? According to Cox postulates, it can be isolated from the host. It can be grown in the media. It can be put back into the new host, and it should show the disease. So here, if your person is suffering from microbacterial infection, you are taking the sample from that animal or organism and trying to put it on the plate. I should see the bacteria growing in. Of course, DNA based methods or sequencing molecular tools that we talk about, they showed the presence of that organism, but it wasn't culturable. It did not grow on the plate. People have tried a number of times over decades to cultivate Mycobacterium leprae, but it remained a big challenge. Even today, even Trypanosoma pallidum, that remained a great challenge. So there are many bacteria, even though the disease caused by them is well studied and is mm, studied with respect to how to control therapeutics are available, but when it comes to cultivation of that bacteria in vitro conditions, it is not really been possible. So there are, even today there are many bacteria like that, and homework for you would be to find out at least two names of pathogens which are not even culturable today. right? So that you can find out and just try to know about the limitations of a microbiologist even in 2020. Now, coming back to the point, what are these molecular tools? Is it that only isolation of DNA, sequencing, and blast? No, of course, there are many more techniques which are referred to as culture independent molecular based methods, and they include denaturation and temperature gradient gel electrophoresis. So, here the word gel electrophoresis is known to you. Gradient gel, so what kind of gradients are there and how are they prepared? Based on that you get the titles as DGGE and TGG, where denaturing agent is used in the grading concentrations, and in another case it is a temperature in the graded system that is used. We are going to learn all these techniques one by one in details. Second is terminal restriction fragment length polymorphism, RFLP. You must have heard about it in basic genetic studies. So that is the technique which is one of the known molecular methods when it comes to culture independent identification of bacteria. 16S rDNA, we talked about it. Then fatty acid methyl ester analysis, that is the total types and composition of fatty acids of a given bacterial cell is going to be unique, right? And you are going to compare it uh, to identify the known bacterial species from the I mean compare it with the known bacterial species and identify the species under study. So fatty acid methyl ester, the what is methyl ester? It is the form which is detectable by tools. So GC chromatography, gas liquid chromatography is the tool. Mass spectroscopy is the tool where you can analyze the typical type of fatty acid present in a given bacteria and match with its family members. Like you match with 16S rDNA sequences in the I'm sorry, 16S rDNA sequences which are deposited in the library. Similarly, you have fatty acid profiles of different bacteria are known and they are deposited. So you can just match the two and find out whether they match or they differ. Of course, in certain cases, this particular tool is not going to be 100% reliable because certain bacteria, certain microbes, yeast, they are going to have different fatty acid profiles when they are grown on different sources. So, what is available as substrate, based on that, their fatty acid profile may change. But that is about storage lipids. When it comes to membrane lipids and other functional lipids, the scenario is not going to be a big challenge. So, fatty acid methyl esters is one of the um, you can say a sample that you are going to obtain from bacteria, that is fatty acid, convert it into its methyl ester form and profile it, right, and match that profile of your isolate with the known profiles and find out the name of your bacteria. And of course, as I said in the beginning, next generation sequencing technology, all these tools we have to learn in details as we progress the progress with the syllabus, right? So you may have to take help of all these different techniques when you are going to talk about study of unculturable bacteria, and of course combined with omics, that is metabolomics, metatranscriptomics, genomics, single cell genomics. All these things will be useful when you are talking about uh, the unculturable bacteria and trying to gain information about them, because it is not as easy that you are seeing a bacteria that is growing in a plate, you are picking it up, purifying it, having an isolated pure culture and studying about it. No, it is difficult. You don't get to see it as a pure colony, right? It is there in a common combined pool. So all of you are together in a classroom. I can identify who is who. But if you are sitting um, on the open ground and all the students of the college are there on the ground, will it be so easy? Of course not. So I'll have to use certain tools to identify and to define who are there from the particular class. Same applies here. You have to have combined approaches combine them with omics also, then only you can get enough amount of information or knowledge about the unculturable, or you can say yet to be cultured. Today we don't get a success to culture them, but it doesn't mean that they are not growing, they are not alive, they are, but we fail to grow them. So we can rename it as, or recall call it as yet to be cultured bacteria. And um, knowing the potential of these bacteria, the pathways they execute, and the kind of special characteristics they have, are they of any application with reference to industrial applications, or are they causing the threat to the particular ecosystem? Are they causing pathogenesis in certain flora and fauna? All these things can be studied by variety of tools, and all these tools we have to learn.